I'm Professor Sims, and in this video I will discuss the tissue level of organization. This is the second in a series of 10 lessons held as part of my Anatomy and Physiology 1 course. If you're a student currently enrolled in this course, please consult the syllabus and course Moodle site for assignments, quizzes, due dates, and other course information. In this unit, we will learn about the main tissue types and their roles in the human body, the 4 types of tissue membranes and the characteristics of each, the functions of various epithelial tissues, connective tissues, muscle tissues, and nervous tissue, and how their forms enable their functions. The term tissue refers to a collection of cells that share a common embryonic origin, exhibit morphological similarities, and are arranged in a specific pattern to perform designated functions in the human body. 4 primary types of tissues exist. These are epithelial, connective, muscle, and nervous tissues. Each type plays distinctive roles important to overall health, and any disruption in their functioning may indicate injury or disease. The origin of tissues traces back to the zygote, developing from totipotent embryonic stem cells that undergo differentiation into the 3 germ layers, ectoderm, mesoderm, and endoderm. These germ layers subsequently give rise to a variety of tissues and organs within the human body. Figure 4.2 gives examples of the main 4 types of tissue in the human body. These are the epithelial tissue, connective tissue, muscle tissue, and nervous tissue. Epithelial tissue lines the gastrointestinal tract organs and other organs. The skin epidermis is also an example of epithelial tissue. Examples of connective tissue are fat, bone, and tendons. Muscle tissues can be skeletal, smooth, or cardiac, which involves the heart. And nervous tissue is of course found in the brain, spinal cord, and nerves. Tissue membranes can be classified as either connective or epithelial. Connective tissue membranes, shown in Figure 4.4, include the synovial membranes that line joint cavities and produce fluid within the joint. Tissue membranes, whether connective or epithelial, have diverse roles in covering external surfaces, organs, internal passageways, and joint cavities. Connective tissue membranes serve to encapsulate organs, whereas epithelial membranes, such as mucous and serous membranes, line body cavities, passageways, and organs, providing protective and lubricating functions. Epithelial tissues are integral for the body's structural integrity. Epithelial tissue also contributes to the formation of glandular tissue. Derived from all 3 embryonic layers, these tissues are characterized by specialized intercellular connections. The cells exhibit polarity with distinct basal and apical surfaces, and their avascular nature relies on diffusion for nutrient supply. Additionally, epithelial tissues possess the remarkable ability for rapid cell replacement. Their primary functions include serving as the body's initial defense against external factors, controlling permeability for selective material transfer, and being capable of secretion, such as releasing mucus. The classification of epithelial tissues involves considering cell shape, squamous, cuboidal, or columnar, and layer arrangement, simple or stratified, with variations like pseudostratified, columnar, and transitional epithelium.
Epithelial tissue is one of the 4 major types of tissue in the body, and it can be found throughout many parts of the body. It lines many of the structures of the respiratory tract, such as the trachea, bronchi, bronchioles, and alveoli, which are the tiny air sacs in the lungs. It also lines most of the digestive tract, the epidermis of the skin, the oral cavity, and many of the glands in the body. Epithelial tissues have some common characteristics. It is comprised of tightly packed cells with very little extracellular space. They also have an apical or free surface that is exposed to an internal body space or the external environment, and a basal or deep surface that is attached to a thin basement membrane which is connected to connective tissue, as you can see here. They also lack blood vessels, which is called avascular, so they get nutrients from their apical free surface or by diffusion across their basal surface from the underlying connective tissue. Epithelial tissue is also innervated, which means that they have a rich supply of nerves, and epithelial cells can reproduce themselves, which is important because they go through severe wear and tear, such as in the skin and the respiratory and digestive tracts. Epithelial tissue has several important functions. It provides protection, as the skin protects the internal body from bacteria and other harmful substances. It provides sensory functions in the skin, nose, eyes, and ears. Epithelial cells are also important in secretions, as they secrete hormones, sweat, digestive juices, and mucus. And epithelial tissue is important in absorption, such as nutrients in the gut and the exchange of gases in the lungs. Epithelial tissue can be classified based on their cell shape or their number of cell layers. Based on shape, they can be squamous, which are flat, wide, and a bit irregular. These are found in the air sacs in the lungs. They can be cuboidal, which are similar to a cube, about as tall as wide, and have a spherical nucleus. Columnar cells are taller than they are wide, and have an oval-shaped nucleus. Pseudostratified cells, as you can see here, have varying heights, as not all of them reach the apical or top surface, but they all do connect to the basement membrane. Some epithelial cells may contain goblet cells, which secrete mucin, which forms mucus that helps with lubrication and protection, and some cells have extensions called cilia, which are sensory organs and also provide movement of mucus away from the lungs and toward the mouth. And some epithelial cells have tiny finger-like projections called microvilli, which is often termed the brush border. Microvilli helps to increase the surface area for digesting and absorbing nutrients in the intestine. Based on cell layers, epithelial cells are classified in 3 different ways: simple, stratified, and pseudostratified. Simple epithelium is only 1 cell layer thick, and all cells are in direct contact with the basement membrane. There is simple squamous, simple cuboidal, and simple columnar epithelium. Stratified epithelium have 2 or more layers of cells. Only the deepest basal layer cells have direct contact with the basement membrane, and their formation looks like a brick wall. This formation provides better structural support and protection from wear or tear. Again, there are stratified squamous, stratified cuboidal, and stratified columnar epithelium. And pseudostratified epithelium is comprised of pseudostratified cells we discussed earlier. 2 other notes regarding epithelial tissue. There are also transitional epithelium, which can be in a relaxed or stretched state. In a relaxed state, they have umbrella-looking rounded cells at the top apical surface, and the bottom basal cells appear cuboidal. In a stretched state, the top cells flatten and appear squamous. Some of these cells will also have 2 nuclei, and transitional epithelium is found in the urinary tract. There is also what is termed keratinized stratified squamous epithelium, in which the top superficial layers of cells are dead. As new cells are produced in the basal bottom region, they migrate to the top, and they produce a protein called keratin. These cells fill with the keratin, which make them very strong, but they lose their organelles and nuclei and die. The epidermis, or outer layer of the skin, consists of keratinized stratified squamous epithelium.
Figure 4.5 illustrates the 3 basic types of cell-to-cell junctions. Tight junctions create a barrier by sealing the space between cells, ensuring selective passage of ions and molecules. Desozones provide mechanical strength by anchoring cells together through intermediate filaments, and gap junctions enable direct communication via channels that allow the exchange of small molecules and ions between neighboring cells. Epithelial tissues are classified based on the shape of their cells and number of layers they form. Squamous epithelium consists of flat, scale-like cells, ideal for facilitating rapid diffusion and filtration. Cuboidal epithelium consists of cube-shaped cells, often found in glands, and they're responsible for secretion and absorption. Columnar epithelium cells are elongated, and they're important for absorption and secretion in tissues like the intestines. The diversity of epithelial shapes reflects their specialized roles in various physiological processes throughout the body. Glandular epithelium, a subset of epithelial tissues, is categorized into endocrine and exocrine glands. Endocrine glands release hormones directly into the bloodstream, influencing various physiological functions. The thyroid gland, for example, produces hormones that regulate metabolism and energy balance. Exocrine glands release their secretion onto the body's surface, often through ducts. Sweat glands, for instance, produce sweat that helps to regulate body temperature and eliminate waste products. The secretion mechanisms of glands include merocrine, epicrine, and holocrine. In merocrine secretion, cells release their products via exocytosis, where vesicles containing the secretory product fuse with the cell membrane, releasing the contents into the extracellular space. Salivary glands and sweat glands commonly employ merocrine secretion for the release of enzymes in sweat. Apocrine glands release their secretions along with portions of the cell cytoplasm. Mammary glands, responsible for milk production, utilize apocrine secretion, where the apical portion of the cell is pinched off, carrying the secretory product. Holocrine glands release their entire cellular contents, including the cell itself, as the product. Sebaceous glands are associated with hair follicles and secrete an oily substance called sebum. Sebum helps lubricate the skin and hair, preventing dehydration and maintaining a protective barrier against external factors. Figure 4.11 shows sebaceous glands connected to a hair follicle. These are holocrine glands that are destroyed and replaced after they release their sebum oil. Connective tissue is so named because it connects tissues and organs, forming a matrix composed of ground substance crisscrossed by protein fibers. Connective tissues support and connect other tissues, provide protection through structures like fibrous capsules and bones, and include specialized cells for defense against microorganisms. Connective tissues are classified as proper, supportive, or fluid. Connective tissue proper is further divided into loose or dense. Loose connective tissue, such as areolar and reticular tissue, acts as a supportive framework, while dense connective tissue, whether regular or irregular, offers resistance to stretching. Examples of supportive connective tissue are bone and cartilage. Fluid connective tissue, such as blood and lymph, transport cells and nutrients throughout the body.
Connective tissues are most abundant and widely distributed in the body of higher animals. They are named connective tissues because of their special function of linking and supporting other tissues or organs of the body. Let's examine different types of connective tissue and their functions. Connective tissues are classified into three types: loose connective tissue, dense connective tissue, and specialized connective tissue. Loose connective tissues have cells and fibers which are loosely arranged in a semi-fluid ground substance. It is further divided into two types: areolar tissue and adipose tissue. Areolar tissue is present beneath the skin and serves as a support framework for epithelium. It contains fibroblasts, macrophages, and mast cells. Adipose tissue is another type of loose connective tissue located mainly beneath the skin. The cells of this tissue are specialized to store fats. The excess of nutrients which are not used immediately are converted into fats and are stored in this tissue. Dense connective tissue are those tissue in which fibers and fibroblasts are compactly packed. On the basis of orientation of fibers, dense connective tissue is further divided into two types: dense regular and dense irregular. When orientation of fiber is regular, in that condition it is called as dense regular tissue, while in dense irregular, fibers arranged in irregular pattern. Specialized connective tissue. Bones and blood are different types of specialized connective tissue. Bones have a hard and non-pliable ground substance rich in calcium salts and collagen fibers, which give bone its strength. It is the main tissue that provides structural frame to the body. Bones support and protect softer tissues and organs. The bone cells or osteocytes are present in the spaces called lacunae. Limb bones, such as the long bones of the legs, serve weight-bearing functions. They also interact with skeletal muscles attached to them to bring out movements. The bone marrow in some bones is the site of production of blood cells. On the other hand, blood is a fluid connective tissue containing plasma, red blood cells (RBC), white blood cells (WBC), and platelets. It is the main circulating fluid that helps in the transport of various substances.
Figure 4.12 shows an example of connective tissue proper, which is fibrous tissue produced by fibroblasts. Notice the individual fibrocytes, adipose, and mesenchymal cells that are fixed within a matrix of reticular, elastic, and collagen fibers. Figure 4.13 gives an example of loose connective tissue with very little matrix. This adipose tissue consists of fat cells that are used by the body for energy and insulation. So notice the difference. There's a lot of fibrous matrix going on with connective tissue, whereas adipose tissue has a very loose matrix. Figure 4.15 gives two examples of dense connective tissues, regular and irregular. Dense regular connective tissue consists of collagen fibers packed into parallel bundles, while dense irregular connective tissue consists of collagen fibers interwoven into a mesh. Cartilage is a connective tissue consisting of collagen fibers embedded in a firm matrix of chondroitin sulfates. Hyaline cartilage provides support with some flexibility, and fibrocartilage is compressible and can absorb pressure. Elastic cartilage provides firm but elastic support. And notice how the structure of these different types of tissue lend directly to the function that the tissues provide. Muscle tissue, vital for movement, exhibits excitable and contractile properties. Muscle cells respond to stimuli and can shorten, generating a pulling force. The three types of muscle tissue are skeletal, cardiac, and smooth, and they differ in structure and function. Skeletal muscles attached to bones enable voluntary movements, constituting 40% of body mass. They generate heat during contractions, contributing to thermal homeostasis. Skeletal muscle cells, or myocytes, derived from myoblasts, form multinucleated fibers with striations. Cardiac muscle, found in the heart, contracts rhythmically and involuntarily. Cardiomyocytes are single nucleus cells, and they're connected via intercalated discs, forming a syncytium for coordinated action. Smooth muscle, responsible for involuntary movements and internal organs, lacks striations and appears spindle-shaped with a single nucleus. It functions in various systems, including digestion, reproduction, and circulation.
First, let's talk about muscle tissue. Muscle tissue is made up of muscle fibers. These fibers are the muscle cells, and they have structure that aids in their function. There are three types of muscle tissue we'll discuss. Cardiac muscle tissue. Like its name suggests, it's in the heart. The muscle fibers are branched and striated, or striped. Each fiber has a nucleus. At the ends of the fibers, you'll find something called intercalated disc. These are really important because they're involved in helping the cardiac muscle tissue contract in an organized, wave-like pattern. This muscle tissue control is involuntary. That is, you do not consciously control it. Smooth muscle tissue. It's smooth, really just meaning here it doesn't have striations or stripes. Each fiber has one nucleus, and each fiber is spindle-shaped, meaning they are wide in the middle and then taper off at both ends. You'll find them in the digestive system, in arteries and veins, in the bladder, in the eyes, changing the iris size. They're also involuntary. Skeletal muscle tissue. This is the one you think about with biceps or triceps because skeletal muscle is what attaches to bone or skin and is involved with voluntary control, meaning you can consciously control it. You can choose to pick up that biology textbook. If you could zoom in to see these skeletal muscle fibers are striped or striated, the fibers are long cylinders that are multinucleated, fancy term for multiple nuclei. All muscle tissue have some characteristics to mention. It can stretch or extend, extensibility. It can retract back to its starting length, elasticity. Muscle tissue also has excitability. That means these cells have the ability to be stimulated. And in the case of muscle tissue, their membranes can have electrical changes and send action potentials. Muscle tissue also has the ability to contract, or contractility. There are some differences in how the contraction happens in the three tissue types. For the rest of this video, we're really going to focus on that last tissue mentioned, skeletal muscle. Let's look a little bit about how they're named, how they're arranged, and how they contract. Many skeletal muscles are named by their location or their shape. Many have Latin or Greek root words in them, so checking out a root word definition list can be really helpful. For example, rectus femoris, it's a muscle on the thigh, or rectus abdominis, that's a muscle on the abdomen. The Greek letter delta looks like a triangle, which is a fitting name for deltoids, a triangular-shaped muscle. There are some beautiful diagrams of skeletal muscles where you can explore the many muscle names and locations. As we mentioned, many skeletal muscles can pull on bones. The part that attaches to the bone that will be moved is called the insertion, and the part that attaches to a fixed part of the bone is called the origin. There could be several muscles involved in a single action. The main muscle doing the work, the prime mover, is called the agonist, whereas antagonists are muscles that do the opposite action, which is helpful for keeping position. If we zoom into some skeletal muscle at the cellular level, how does it do what it does? How does it contract? This is probably the most exciting part of the video. Okay, so imagine you have some skeletal muscle like the biceps muscle. That muscle is made up of many muscle fibers, which remember are muscle cells. By the way, some of these cells are big. Of course, generally cells are microscopic, but these muscle cells can have lengths up to 30 centimeters long. Anyway, inside a muscle fiber are multiple myofibrils, which are long cylinders. Each myofibril has sections that repeat called sarcomeres. And it's the arrangement of these sarcomeres that contribute to the skeletal muscle's striated look. There's a lot to a sarcomere, and that's where we're going to focus. So the sarcomere has a protein known as actin. Actin makes up what is known as thin filaments. The sarcomere also has a protein called myosin. Myosin makes up thick filaments. To try to remember which one is which, I try to remember that the word thin is almost in the word actin, minus the H though, but close enough to help me remember. Anyways, both of these are essential in causing muscle contraction. Enter the sliding filament model of muscle contraction. Now, as we often tell you, we're going to do a simplified version. When you're ready for more to explore, check out our video description. Okay, so we have actin, thin filaments. We have myosin, thick filaments. We have these Z lines here where a sarcomere ends, and these Z lines are where the thin filaments attach. The thick filaments are held by accessory proteins in this area called the M line. The big, super important concept here is this. The sarcomere must shorten for muscles to contract. But the thick and thin filaments themselves do not shorten. So the way that is going to happen is that we're going to slide past each other. And they do. When the sarcomere contracts, the thin filaments will be pulled by the thick filaments towards the center. There is overlap of the thin and thick filaments. Z lines will be moved closer together.
This table describes the 3 types of muscle tissues in terms of histology, function, and location. Skeletal muscle cells have prominent striation and nuclei on the periphery. They allow for voluntary movement, produce heat, and protect organs. Skeletal muscles attach to bones and entry points like the mouth and anus. Cardiac muscle cells appear striated and have a single nucleus. They are found in the heart and contract to pump blood throughout the body. Smooth muscle cells have a single nucleus and no visible striations. They're involved in involuntary movements like breathing and dilation of arteries, and they're located on the walls of major organs and passageways in the body. Nervous tissues transmit electrochemical signals facilitating information exchange within the body. Nervous tissue comprises 2 primary cell types, neurons and neuroglia. Neurons employ electrochemical impulses, known as action potentials, which are linked biochemically to the release of chemical signals for propagating information. Neuroglia support neurons and modulate the propagation of information. Neurons exhibit distinctive morphology featuring 3 main parts: the cell body, dendrites, and an axon. Neurons are categorized as multipolar, bipolar, or unipolar based on their dendrites and axon configurations. When successfully stimulated, neurons generate action potentials that propagate down the axon toward the synapse, leading to a response if enough neurotransmitters are released to stimulate the next neuron or target. Astrocyte cells, distinguished by their star shape, are abundant in the central nervous system. They perform various functions, including the regulation of ion concentration, uptake and breakdown of neurotransmitters, and the formation of the blood-brain barrier, which is a membrane that separates the circulatory system from the brain. Microglia protect the nervous system against infections, although they're not technically considered nervous tissue since they're more related to macrophages. Oligodendrocyte cells produce myelin in the central nervous system, and Schwann cells produce myelin in the peripheral nervous system.
The neuron is the basic unit in the nervous system. In a simple animal like a nematode worm, they only have 302 neurons, but what they allow them to do is to take information in from their surroundings and respond to that information. If you look at how those 302 neurons are connected, it's very complex. Now, humans are quite a bit bigger, and we have 100 billion neurons in our body. Most of those are centered in the central nervous system, in the brain, and the spinal cord, but the basic unit of our nervous system is the same. It's the neuron. If we look at the neuron anatomy and remove all the other neurons, the center is going to be what's called the cell body. This is a typical cell. It's a metabolizing cell. It's taking in energy. It's using that energy, and so it's going to have a nucleus. It's going to have Golgi apparatus, mitochondria. It's a typical cell, but there are extensions of those cell bodies called the dendrites. Those dendrites are connected to other neurons, and they're taking information in the form of neurotransmitters. In fact, this whole side of the neuron is receiving information. We then have the axon. The axon is where we're transmitting information. It begins at the axon hillock and is going to move to the terminals. Now remember that the terminals, there's a synapse that connects it to other neurons, but the signal is going to move in this direction. We're taking in information from the dendrites, the cell body, and then we can send information to the next neuron. Now how do we do that is through an action potential, and I have a video that I'll put a link to where I go into the specifics of how that occurs. But certain axons in our body are really, really long, and for that information to travel quickly, we will wrap them in myelin sheaths. It's a Schwann cell that's wrapped around it. The way it works is we put the voltage-gated channels in between that, and it acts to insulate that nerve. It allows the information to travel more readily down that axon. And we generally say if you have an axon that's wrapped in myelin sheath, we call that a nerve fiber inside our body. Now we're taking in information as well. So there's going to be another neuron that's connected to both the cell body and the dendrites of that cell. But there's going to be a gap between these two, and we call that a synapse. It's a connection between neurons. Again, I have another video on the synapse if you want to dig into the specifics, especially of how chemical synapses work. Neurons are classified in two ways. First way is due to their structural classification. So if we take the cell body of a typical neuron, like this multipolar neuron, the way you can figure out what type of a neuron is find the cell body and figure out how many poles are coming off of that cell body. So if we count them, we have a number of different dendrites that are coming off of this neuron. And then we have one axon that's coming off. And so we call that a multipolar neuron. This is the most common neuron that we have in our body. If we look at the next one, we find the cell body again. How many poles do we have coming off of it? We've got the axon coming out and the dendrite coming in, and so we call this a bipolar neuron. Bipolar neurons are very rare. They transmit information from the receptors in our eye, receptors in our nose. If we look at the next one, here's the cell body. You can see there's only one pole coming off of that. It's actually a fused axon and dendrite, and we call that a unipolar neuron. Now, in vertebrates, it's sometimes referred to as the pseudo-unipolar neuron. These are going to be most of the sensory neurons in our body. They're taking information in and transmitting that back into the central nervous system. And then if we look at the next one, if we look at that, you can see there's no axon on it at all. And so we call that an anaxonic neuron. These are found in the brain, and they're not super understood. Now, there are a lot of other structural classifications, but these are the general ones that you're going to find in a typical anatomy class. Now, neurons can also be classified according to their functional classification. Those that take in information, stimulus, coming in from our environment, and then the response that we have. So if we kind of move that to the central nervous system, we're looking at a cross-section of the spine. So we're taking information in from receptors. So these could be in the skin, for example, touch receptors, heat receptors, pain receptors. And so we have to transmit that information to the central nervous system. So those neurons that bring information in are sometimes referred to as sensory neurons or afferent neurons. You can see this is a unipolar neuron, and it's transmitting that information into the central nervous system. And then we have information going back out. We're sending information out to effectors in our body. It could be muscles, could be glands, and we're going to call those motor neurons. They're moving away from the center. Sometimes we refer to those as efferent neurons as well. Now, there should be connections between those neurons. Those aren't all the neurons we have in our body. And so we also have these connections in the middle, and those are going to be called interneurons.
Figure 4.19 illustrates three main parts of the neuron: the cell body, also known as the soma, the dendrites, which carry the action potential, which transfer nerve impulses to the soma, and the axon, which carries the action potential away from the soma and toward another cell body. Figure 4.20 shows the nervous tissues with the neurons and their components. So we have the oligodendrocyte, myelin sheath, astrocytes, microglial cell, and then the neurons themselves. Tissues are susceptible to injury and aging. During tissue injury, inflammation serves as the initial response. Acute inflammation helps to limit damage and initiate tissue repair. Chronic inflammation can lead to conditions like arthritis. The four cardinal signs of inflammation are redness, swelling, pain, and localized heat. Tissue repair involves clotting, scab formation, and fibroblast-driven regeneration. Figure 4.21 helps to illustrate what happens during tissue healing and wound repair. Initially, there is inflammation where blood vessels constrict to minimize bleeding. This is followed by the release of inflammatory cells that clear debris. Then, the fibroblasts deposit new collagen, and the wound undergoes remodeling, gradually restoring tissue strength and function. Aging impacts all cells, tissues, and organs, and results in visible changes and increased susceptibility to disease. While external signs of aging are noticeable, internal signs such as slower wound healing and increased susceptibility to disease are equally significant. At the cellular level, aging involves telomere shortening, altered cell membrane function, and the potential development of age-related diseases. Cancer, a result of uncontrolled cell growth, invasion, and colonization, is driven by mutations or epigenetic modifications. Tumor formation, whether benign or malignant, reflects abnormal cell division. Cancer treatments, including surgery, radiation, chemotherapy, and targeted therapies, aim to eliminate or control cancer cells, each with their own limitations. Researchers are exploring pharmaceutical targeting of specific proteins and cancer-associated molecular pathways for more effective treatments. Cancer development typically begins with hyperplasia, an abnormal increase in cell proliferation. This leads to the formation of an enlarged tissue. If unchecked, this can progress to dysplasia, where cells exhibit irregularities in size, shape, and organization. As the transformation continues, the next stage may involve the formation of in situ tumors, where abnormal cells are confined to the original tissue, but have the potential to invade neighboring structures. If the cells acquire the ability to invade surrounding tissues and spread to distant sites, the tumor becomes malignant. Malignant tumors can be further classified based on their tissue of origin, with carcinomas being cancers arising from epithelial cells, which are the most common type of carcinoma.
The microenvironment has always fascinated me. I was not even in grad school yet, and I heard Mina Bissell give a talk where she showed a cell that she had taken from the prostate and put in a mammary microenvironment, and that cell actually started producing milk, which I thought was the most fascinating experiment I'd ever seen.
Professor Ashani Weeraratne has been studying the cellular microenvironment in her lab for the past 17 years. Cells don't live in isolation. The other cells, molecules, and blood vessels in their surroundings can have a powerful effect on their behaviour. And this is especially important when it comes to cancer.
So the tumor microenvironment is really the neighborhood in which the cells sit. They sit in a network of matrix and mesh, and they have other cells that are around them, and the impact and the crosstalk of those cells with the tumor cells can have a huge impact on the behavior of the tumor cells outside of genetics. My lab and others are looking at the impact of aging of normal cells in the tumor microenvironment and how those impact cancer development and progression. It's such an obvious thing that cancer and aging go hand in hand. And yet, despite that, until we really started digging around, I don't think even I realized how important it would be to understand what those changes are and how we can target them.
Cancer is often defined as a disease of aging, and the vast majority of cases occur in people over the age of 50. As global populations live longer, cancer is set to become a major public health burden. So how does an aging microenvironment around a tumour affect cancer progression?
What we're finding is that the cells that are aging normally as they're supposed to do around the tumor cells are sending signals to those tumor cells that make them more metastatic and more invasive and more resistant to therapy. For example, if you look at the skin, we see collagen that is nice and firm and very dense in younger skin, but that we lose that density and that firmness in aged skin. And that provides ways for tumor cells to move differentially through that collagen. Also, immune cells. The immune system changes dramatically as we age. And what we're finding is that there are changes that are both beneficial as well as detrimental in the aged tumor microenvironment. So, for example, in melanoma, patients are often treated with therapies that boost the immune system, so immunotherapies, and we find that older patients actually respond much better to immunotherapy such as anti-PD-1 than younger patients because they don't have the same level of regulatory checkpoints that younger patients tend to have. So one of the things we've been most excited about recently is the fact that we have found changes that occur in tumor cells that we put into age microenvironments rather than young microenvironments. And we can actually target those changes. We see those changes in patients, and when we target them in mice, we can overcome therapy resistance and reduce metastasis. And so that is fascinating, and we're hoping to push that into clinical trials over the next few years.
So are researchers currently paying enough attention to the effects of the aging tumor microenvironment?
As a community, we're not really taking the aging microenvironment into account. We are designing preclinical studies that are all based in 6 to 8 week old mice. We don't really tailor our therapies from young to older patients. Our work has revealed that older patients respond very differently, even to targeted therapy. So that has been a really fascinating finding for us, because one of the early experiments that we did was to take genetically identical tumors that bore a mutation that we could target. And yet when we put the same tumors in either a young mouse or an aged mouse, what we saw was that the tumors in the aged mice, despite the fact that you had one gene and one drug to target that gene, behaved completely differently. I think one of the most important milestones we're going to see over the next 5 to 10 years is the movement of some of this work into the clinic. So for example, the target that I was speaking of earlier is a target that we can make a drug for, which we are doing, and then get into patients in the clinic. I also hope that people will start thinking about designing preclinical studies in young and aged mice. We're beginning to show that there are differences in older females and older males. So I think that's going to be an area of great importance as well. And finally, really what I'd like to see is the way we treat patients, how important it is to treat them according to their age.
Thank you guys for watching. Don't forget to do the reading, check the descriptions for more videos related to these topics, and leave your questions for me in the comments below.